folks. So this is part one of a two-part lecture series regarding evidence for evolution. So in this lecture series, I'll be kind of discussing and considering the different indications or traces of evolution. In other words, what evidence exists, or all the different lines of evidence that exist that kind of show evolution has taken place in the past and is still occurring today. Now, I like to say the concept of evolution encompasses three kinds of questions. The first one being the fact of evolution. So, what evidence is there that proves evolution did occur? And we'll be discussing these different lines of evidence. The second is the course of evolution. So, did it occur at a small scale or was it a large scale? And then that ties in with the third, which is the mechanism of evolution. So, how did evolution actually occur? Was it through the operations of natural selection? Was it a product of genetic drift? Kind of that aspect. How did it actually occur? So looking at how do we know it occurred? What was the duration of it, or what time scale did that occur? And how did it actually occur? If that makes sense. So evidence for evolution comes from many different areas of biology, the major ones being these five. So first is the fossil record. Fossils document the existence of now extinct species that are related to present day species. There's biochemical, or also known as molecular evidence. That is referring to DNA and the genetic code. Both of these reflect the shared ancestry of life. And oftentimes we use DNA to compare exactly how related species are to one another. There's comparative anatomy. That's kind of my favorite, but my opinion is a bit biased as a homologist and lover of all things dealing with bones and remains and such. But comparative anatomy, so oftentimes with this, we see species that may share similar physical features because that feature was present in a common ancestor. Also go over how this isn't always the case. There are observable events, or direct observation. So this is particularly true of organisms with short life cycles. If you consider pesticide resistance in insects, it's very easy to observe small-scale evolution at this level, where you can actually see insects evolving very quickly to have pesticide resistance. The same holds true for antibiotic-resistant bacteria. Often these bacteria will gain some sort of antibiotic resistance through various modes of horizontal gene transfer, which is when you have unrelated organisms kind of transferring genetic material between each other. The primary mode is something known as transformation, and that's where you have competent bacteria taking up naked DNA from the environment, and sometimes they'll take up plasmids or DNA material from the actual bacterial chromosome that has some sort of advantageous phenotype associated with it. So you see some bacteria gaining a phenotype that allows them to have antibiotic resistance, or they gain degradative plasmids, which are these plasmids are essentially extra chromosomal DNA. Degradative plasmids allow bacteria to gain the ability to degrade environmental pollutants or toxins. So again, those are observable events where you see evolution occurring on kind of this small scale level. The last one is biogeography. Biogeography pertains to the global distribution of organisms and the unique features of island species. Both of these not only are reflective of evolution, but they also reflect geologic change. So we'll begin with the first area of evidence, looking at the fossil record. Now, what are fossils? Well, the term fossil refers to any part or impression of an organism that can survive or may survive long after its death. In other words, fossils are the preserved remains or traces of previously living organisms. Usually they'll form best when an organism is buried quickly in conditions that kind of slow the process of decay. Oftentimes we find fossils preserved in sedimentary rock, and in the next slide we're going to get into the different types of fossils, but what actually constitutes as a fossil differs in a lot of cases. So very rarely do you have the preservation of organic matter. That occurs in rare instances, often with plants when they're kind of compressed between layers of stone, but rarely do you have soft tissue preserved. Oftentimes the soft tissue will be replaced by minerals that are dissolved in the water, and it basically forms them into stone. Or you'll have the preservation of mineral-rich hard parts, which are the bones, teeth, and shells. So again, it varies. Sometimes you get molds or different casts, and we'll go into that in a moment. Now, the fossil record that we have at this time is incomplete. It's not unbroken, it's not complete. And there are several reasons for this. I had somebody ask me one time, why is the fossil record incomplete when there are so many fossils out there? And it's a valid question, but there's four major things to consider. One is that, again, soft tissue rarely fossilizes. Secondly, some organisms will never fossilize. Oftentimes, fossils will only form in certain environmental conditions or in certain habitats. So there's that potential chance that an organism will not fossilize. You also have to consider that the movement in the Earth's crust and the tectonic plates, sometimes that will actually cover or push deeper the layers containing fossils. It can also destroy fossils, and if it's a large enough shift in the plates, they can also redistribute the fossils, so they can kind of almost scatter them per se, or drift them further apart so that it's more difficult to actually find a complete organism. And also, we haven't dug up every place on this globe. There's a lot of areas that are still untouched by paleontologists, and it's going to remain that way for a while. So we can't uncover every area of the Earth, unfortunately, but that's the reality of it. And natural events, again, can either expose different fossils or can actually recover and more deep recover fossils. But the fossils that we have found or that we have collected over time, they have provided unique insight as to evolution over long timescales. You're probably wondering, well, what are these insights specifically? Well, because of fossils, we've gained information regarding extinction events. The fossils have shown that different organisms have appeared and disappeared throughout geologic history. Fossils have also provided crucial information regarding transitional forms. So transitional forms are species that kind of link groups together. Some classic examples would be Archaeopteryx. That is this bird-like reptile, per se. It looks like a lizard with wings, and that serves as a link, or that's the transitional group linking avian species or your birds with early reptiles. There's Eusopteron. Those are amphibious fish. So they're one of the three kind of transitional groups that link the transition from early fish to your first terrestrial amphibians. There's also Simoria, if I'm correct in the name. Those are reptile-like amphibians, and so that group provides a link between your early terrestrial amphibians and the first early reptiles. While fossils have provided a plethora of information regarding species development, they've also provided valuable information regarding primordial ecosystems. Primordial meaning ancient or kind of ancestral origins, per se. So very early life. From this, the fossils have revealed ancient climates, different environmental conditions. It's also through fossils that we've learned about the development of life from single to complex, and that life began or originated in water and later transitioned onto land. All right, so there are 5 types of fossils. Imprints, that's the first group. That pertains to carbon films and impressions. These are formed when you have the escape of liquids and gases from an organism as it's compressed between layers of soft stone. So a beautiful example is this picture here. So these are the kind of the carbon imprint of fern fronds, and those were created as the ferns were compressed between layers of shale, and the different gases and liquids escaped, and what you're left with is basically a picture of the original organism imprinted onto that ground substance. There are molds and casts. Mold and casts are essentially replicas, hardened replicas of the organism. You can see this example here. This is a mold and cast of a shell. And the nice thing with the molds and your casts are that oftentimes you have the preservation of very fine detail. You can actually see all these striations preserved here in the mold. A lot of the molds form initially underwater. So underwater, as sediment kind of fills in and around the dead organism, it creates a mold. You can think of that as an outer shell. And then over time, as that actual organism decays, the space that it originally occupied will fill with sediment. And that's what creates your cast. So again, they're hardened replicas of the space once occupied by an organism. Petrification is the third. So if you've ever been to the petrified forest, you've seen these petrified fossils. You go and you find these kind of slabs of wood and different parts of trees that look like wood, but then you go and pick it up and it's extremely heavy and hard, and it's literally stone. The reason is because with these fossils, with petrified fossils, original tissues are replaced by minerals that are dissolved in water, and they'll replace the organic matter, and that process essentially turns it into stone. You can also have, again, the preservation of mineral-rich hard parts, which are your bones and teeth, etc. That's what's up here. You have the preservation of bones. With oftentimes with that, minerals will replace the calcium phosphate in bones and kind of harden it that way. Let's see, there are original remains, which are also known as true form fossils. These are these three up here. So true form fossils are when, or pertains to when you have an entire organism preserved or fossilized in some sort of hardened substance. And oftentimes we see that substance being amber, tar, or ice. So here is an example. There's two insects that have been fossilized in amber. When they're fossilized in amber, that means they're preserved in tree sap. So this is a hard substance, and it just kind of envelopes the entire organism and then hardens and preserves it in that manner. This is one of the few cases where you have the preservation of actual organic matter and sometimes even soft tissue, which we have over here. So this is a 100,000-year-old mammoth from Siberia that was preserved in ice. So because of that flash freeze, and because it remained frozen for thousands of years, it actually preserved a lot of the flesh and even some of the fur. And if I'm correct, their organs still remain. They're desiccated, but everything is well preserved. This one over here. So this is the kind of the skeletal remains of the fossilized skeleton of a 2-meter high giant ground sloth, and that was preserved in the tar pits in California. The last group are what we call trace fossils. So trace fossils don't involve the preservation of the actual organism per se. Rather, you have the preserved remains of past activity of those organisms. So you have the preservation of footprints or different trails, nests, burrows. Right here you see different little worm trails that have been preserved in siltstone. So it's the preserved activity. It shows where they've been and moved. It's not preserving parts of the actual organism itself. And I know some of this is review for people, but I always like to go over fossils just because some people have gone over them greatly in their younger educational years. Others have not. So I like getting everybody on the same page. So I'm sorry if this is review, but you can just, if it is, you can feel free to skip ahead to the next kind of section in the video. But in order to have fossils form, obviously you need an organism to die. So the organism dies, and then that carcass will move into the burial phase. During the burial phase, or what I refer to as the kind of the sediment phase, the animal begins to become buried by different layers of sediment. This can be volcanic ash or silt, and that protects the bones from rotting. And then over time, more and more layers will kind of accumulate on top of those remains. And different minerals such as silica, which is a compound of silicon and oxygen, will slowly replace the calcium phosphate in the bones. So this is where you start getting mineralization, and those remains, the actual organic tissue, will start to decay, but minerals fill in that space to create a 3D model of the fossil. This is where you can also have impressions start forming. So with the fern fronds, for example, the gases and liquids start to escape, and it makes an impression of the original remains or the original organic matter on the soft stone or the layers. In time, some fossils can actually be pushed closer up to the surface of the Earth by changes in tectonic plates, or those giant earth plates that move around and cause earthquakes. So they're pushed kind of closer up to the surface, and then over time you have the wearing away of different sediment layers, either naturally by processes of erosion from wind, rain, or different rivers, or the fossil is exposed because of human activity. Sometimes it's found purposefully by a paleontologist, or maybe even not purposely, or you hear about an industrial park that was going to be made, and then all kind of building activity and construction had to be halted because they found either fossilized remains or the remains of a past culture or settlement. All right, so when you find a fossil, how can you actually determine the age of it? And there's kind of two main methods. The first deals with stratigraphy. Stratigraphy is when you place fossils in relative age sequence. Oftentimes, fossils are contained in rocks that build up in layers called strata. And the strata provide a sort of timeline, per se, wherein the layers that are closer to the top or the surface of the Earth are younger. They're newer layers. And then they increase, those strata layers increase in age as you move closer to the bottom. So these layers up here are the youngest, newest layers, and then these down here are the oldest. So they increase in age as you move downwards. And based on that notion, you're able to start figuring out how old a fossil might be based on the depth of the strata, or their position, their ordered position in the strata, and then the depth at which they're found. So for example, looking at these two questions I put here, so we know that fossils A and B were alive at the same time because they're contained within the same strata layer. The oldest fossil is fossil C because that is contained in the bottommost strata layer of that grouping. Another great resource are something called index fossils. So index fossils are used to actually define geological periods. This is where you can actually start getting a numerical value for that age, rather than was this fossil more recent or was it more ancient in geologic history. This allows you to start determining a number. Index fossils are often defined as commonly found, widely distributed fossils that are limited in time span. So they either existed for short periods in geologic history, or they existed only during one geologic period. And that kind of key position within the relative timescale allows them to serve as guidelines for aging the rocks in which they're found. So if you find an index fossil in a layer, you have kind of bounds for determining the age of that layer. Index fossils are also used to correlate strata from different sites. So the example I have here, so these are two different sections from two different sites. So this is section A from site 1, this is section B from site 2. And you're going through and you find kind of this shale layer at both sites. Now both sites have shale layer. Okay, that's great. It doesn't necessarily mean that those two layers are from the same time period. But if you look at the fossils, both layers contain the same fossils. So because they contain the same fossils, that starts giving you an idea that both layers occurred during the same time. Looking closer at the fossils, these are both Ordovician index fossils. So these are two fossils that are specific to the Ordovician time period in geologic history. After, I think it's after the Paleozoic, it's actually, I think even before Paleozoic, you no longer see these organisms. So they're really tied to that moment in history. So, okay, you know that both of these shale layers are from the same time. More specifically, they're from the Ordovician time period based on the fossils they contain. Now, with these index fossils, you can actually determine that the age of the shale layer that they're contained within must be between 450 to 480 million years old, because that's the time or the age of the Ordovician period. So that's kind of how that works. Sometimes you'll find multiple index fossils across different sites. With that, when you want to kind of determine what you're using for an index fossil, you want to make sure that it is kind of found in a layer that's all the way, that's maintained across all sites. So it kind of has that wide distribution, but it's maintained in a relatively short time period for that site. So, for example, you want to see that it's only in one layer. You don't want it to be in like 5 different layers, and you want it to be found in all samples. So that's why here they're using this little shell as kind of the index fossil. So sometimes index fossils may not be what I commonly refer to as index fossils, which are these guys that are key to actual geologic periods. Sometimes they might be, again, one that's coined as an index fossil because you're able to tie it across different locales based on these criteria. Other times you'll have multiple index fossils, but you choose the best one based on this criteria. Another method for dating fossils is radiometric dating. So that deals, or that process measures the radioactive decay of certain elements. In other words, it's measuring the decay of isotopes. When you have an unstable radioactive isotope, that's called your parent isotope. That will decay over time in an effort to become more stable. When it decays, you get two products. One is known as the daughter, which is comprised of the new atoms that are formed from the decay, and those are more stable. And you'll also have radioactive particles that are released. It's either alpha, beta, or a gamma particle. So you can actually measure the proportion of a radioactive isotope relative to its more stable form to kind of determine how much time has elapsed. And more specifically, what you'll use is something known as a half-life. Isotopes decay at a known rate, and that's called a half-life. Half-life is the amount of time that's needed for half of a sample of radioactive material to decay. So you can actually calculate the age of a rock using the formula time equals duration of half-life times number of half-lives that have occurred. This is a chart here of the common isotopes that are used in radiometric dating. A lot of times you hear about carbon-14 used. There's also uranium-235 or 238 and potassium-40. And then you can see what atoms they actually decay into. So carbon will decay into nitrogen, potassium decays into form atoms of argon, uranium decays to form atoms of lead, and then these are the half-lives. So in order for half a sample of uranium-235 to decay into lead and whatever radioactive particle is released, it takes 710 million years. That's just one half-life. Now, the rate of, or the ratio of parent to daughter isotopes change with the passage of time. Changes with the passage of each successive half-life, where the actual number of parent isotopes decrease and the number of daughter isotopes increase. And your parent isotopes will decrease at a rate that's so that you have half remain for each successive half-life. It's the amount that remains is half of the amount of the previous. So you can kind of use that ratio to figure out how many half-lives have actually passed when you're calculating your formula. Hi, folks. Hi. All right, so moving into from fossils into our second area of evidence. This is biochemical evidence or your molecular evidence. Similarities between biological molecules can reflect shared evolutionary ancestry. At the most, kind of if you take a step back and look at the most basic biochemical level, all living organisms share the same genetic material, that's DNA and RNA, the same or highly similar genetic codes, the same basic processes of gene expression, so transcription and translation, and they also share the same molecular building blocks, such as amino acids. And these shared features suggest that all living things are descended from a common ancestor. And this common ancestor that they're descended from had DNA as its genetic material. It used the genetic code, and it expressed its genes through transcription and translation. Now, Figaro, stop it. Now, while these features are great for establishing the common origins of life, some of them, in particular transcription and translation, are not really helpful when we want to determine or figure out how related species are. So when we want to do that, when we want to determine relatedness, or what organisms in a group are more closely related, we have to use different types of molecular features, in particular looking at nucleotide sequences of different genes. And the whole kind of notion behind this when we use the nucleotide sequences is that the more closely related organisms are, the more similar their biochemical makeup will be. So if two organisms are very similar to one another or very closely related, they would have less amino acid or nucleotide substitutions in their sequences. So they would be their biochemical makeup would be very similar to one another. One of the major homologies, per se, that exists for this notion of a common ancestor for all of life really stems from the universality of the genetic code. When we talk about the genetic code, we're talking about the actual underlying makeup, the molecular building blocks, the foundation of genes and how genes are encoded. All organisms have a genome, right? A genome it contains all of the genetic materials of that organism, all of its genetic makeup. And genes, I like to describe them as these unique sequences of RNA that encode for either proteins or a unique RNA molecule. A lot of people forget that there are many genes that do not encode for proteins. The geneticist in myself must remind you that there are some genes that will instead encode for different molecules of RNA, such as tRNA or rRNA, even mRNA. So it's important to remember that distinction. But in the case here, with your genes, when you look at genes across all organisms, their foundations are similar or the same. They all use the same nitrogenous bases to kind of create the nucleotide sequences for the genes. With RNA, these nitrogenous bases are adenine, guanine, uracil, and cytosine. With DNA, it's adenine, guanine, thymine, and cytosine, and they all follow the same base pairing rules. Taking it a step further, these nucleotide sequences in all organisms are grouped in triplets, and those triplets are called codons. Now, each codon codes for a specific amino acid, and those codes are actually well maintained in almost all different species, except for a few different microbes. But almost all species will maintain this genetic code, as we say. So these are the RNA codons. Again, your triplets of RNA nucleotides. Each one codes for a specific amino acid. Now, all proteins are created from the same 20 different amino acids. So there's another kind of homology, the similarity in RNA, DNA, and your proteins. Now, with the genetic code, it's interesting because you'll see redundancy where you'll have the same amino acid coded for by multiple codons. You also see this interesting case of wobble base pairing, which is when the first two sites of the triplet are the same, but can be a variation at this third site. But even with that variation, oftentimes you'll still get the same amino acid. Now, that's not always the case, but you see it a lot. The majority of them, again, maintain, even if you have the first two nucleotides here, you can have a variation at that third position and still have the same amino acid coded for. There's also universal start codon, which is AUG in RNA, and that codes for the amino acid methionine. You see that again across all organisms except for a few different microbes and a couple bacteria, I think it is. But again, it's this universality in the genetic code that really provides a lot of support for evolution, and the similarity of the triplet DNA codes that actually make up amino acids, that also shows a probable common ancestor for all life on Earth. Biologists often compare the sequence of homologous genes in order to determine relatedness. Homologous genes are related genes that are found in different species. And the basic idea behind this is that these quote-unquote same genes are found in a variety of different species because they all descend from a common ancestor that had that gene. So because their last common ancestor possessed that gene, or because that gene was already present in their last common ancestor, all those species have it. A great example is looking at the gene that encodes the hormone insulin. So insulin is not only found in humans, it's found in chimpanzees, cows, dogs, rats, a wide variety of different organisms. And again, the idea behind that is that that gene was already present in the last common ancestor for those species. Now, scientists will use these protein homologies to kind of start creating phylogenetic trees, or to get an idea, again, of relatedness. For determining relatedness, kind of the mechanism that you operate on is the idea that the more DNA differences there are in the sequence for the homologous gene, or the more amino acid differences there are in the proteins that they encode between different species, the more distantly related those species are. So if you have a lot of similarities between their amino acid sequences for the protein they encode, or in their nucleotide sequences, then they are closely related. They are very similar. So using our insulin here, when you compare the insulin proteins between humans and chimpanzees, they are very similar. They're about 98% identical. And that's quite different than when you compare the insulin proteins for humans and chickens. There, it's only about 64% identical. So using that information, you can kind of conclude that humans and chimpanzees are more closely related to one another than humans and chickens. With most proteins, changes appear to accumulate at a constant rate. Now, this is looking at certain proteins in particular. It's not looking at proteins across the board having this universal rate at which they all operate. There's a rate that seems to be specific for example, insulin. There's one that seems to be specific for albumin. So it's different types of proteins, but those proteins seem to have changes that accumulate at a constant rate across a variety of species. And this phenomenon is referred to as a molecular clock. Now, scientists will use molecular clocks to measure changes in DNA or protein sequences to indicate degrees of relationship among species. The one that's most commonly used is a protein called Cytochrome C. Cytochrome C is an ancient ubiquitous protein, and I say it's ubiquitous because it's found in all aerobic or oxygen-breathing organisms. It's found in the mitochondria and thought to kind of serve an essential step in the production of cellular energy. A lot of scientists believe that it actually plays a crucial role. I think it's been found that it plays a crucial role in the electron transport chain during cellular respiration. Now, this protein has changed very little over evolutionary history. And we're talking millions of years of evolution. It's changed very, very little. Human cytochrome C is comprised of approximately 104 amino acids, if I'm correct, and 37 of those amino acids, regardless of the species tested, are found at the same position in all sequenced cytochrome C molecules. So what that means is that those positions, those amino acid positions, are highly conserved in the molecule across different species. And looking at the whole molecule, again, as a whole, a large amount of it is, again, highly conserved across a wide variety of species. So this strongly suggests that this protein, or these proteins in different species, have descended from an ancestral cytochrome C molecule that was found in some primitive microbe. And it's actually believed that this microbe existed more than 2,000 million years ago, which is pretty fascinating. But cytochrome C will be used to create molecular clocks, again, going back to the same concept or same idea that I talked about before, which is where the amino acid sequence of or used to make that cytochrome C molecule will differ increasingly the more distantly related two organisms are. So if those two organisms have very similar amino acid sequences for their cytochrome C molecule, then they're closely related. You can actually look at this table here. So this is looking at the nucleotide differences rather than the amino acid differences. But it's comparing different organisms, and they're all given a kind of a numerical value, which is maintained across the horizontal axis as well as the vertical axis. So in this box here, you're comparing humans with monkeys. Monkeys are listed as number 2. And that shows that there's only one nucleotide difference. Now that one nucleotide, that's just one nucleotide base. Now that doesn't necessarily mean that's going to have an amino acid difference in the sequence. That's where kind of looking at the actual amino acid sequence comes into play. But based on that information, they're virtually identical. And that's quite different than if you look at human versus pig. So pig is number 6. So that's this box right here. They have 13 different nucleotide bases. So that's how they're starting to build this tree over here. The greatest number of differences between humans and Candida, which is a type of yeast. There are 66 different nucleotide base differences. And so the less differences there are, the more closely related. That's why you have pigs here, followed by ducks and snakes, and then tuna, and then moth. They show the number of bases. So the base substitutions will increase the more distantly related they are. All right. Third kind of line of evidence, and my favorite, is comparative anatomy. Comparative anatomy is the study of anatomical structures and involves comparing the structural features of related animals to ascertain the degree of similarity between them. Oftentimes, if species share similarities in structures, it may mean that they share a common ancestor. But again, this isn't always the case, as we'll see. Body structures that are similar in structure due to a shared common ancestor, but differ in function, are known as homologous structures. Kind of the classic example are four-limb structures in dolphins, humans, horses, bats, birds, and whales. Looking at them firsthand, structurally they look different, right? So this forelimb of the dolphin looks quite different from the human, but that's because they are adapted for a different function. These animals evolved in different environments or under different selection pressures, so that's what these structures are reflective of as far as their function. So for dolphin, the forelimb is used for swimming. Humans, it's used for grasping. Horses for running. For bats and birds, its function is flight. So again, they look different because they formed under different selection pressures. But if you take a closer look at the actual pattern of the bones forming that forelimb, you see that they all contain the same complex bone structure, right? So they all have a humerus. There's a radius in all of them. All of them have the ulna, carpals, metacarpals, and then phalanges. Now, some of them, the actual layout of the phalanges is different. So a lot of them are pendactyl, meaning they have that five-fingered kind of layout. Right? You see that with the turtle, human, you even have that in the dolphin, right? From the outward appearance of the fin, you didn't think that they would have five phalanges, but they do. Bats do as well, in their wing. For horse and bird, they still have phalanges. It's kind of modified. They're not pendactyl. With horse, they've fused, a lot of the phalanges have fused together with phalangeal 3 to form the hoof. And then the fourth phalanges has actually receded back below, right below the knee to kind of serve as a support structure. Similar with the bird, so you have the fusion of metacarpal 1 and 5 with 3 to create this kind of pointed phalangeal unit here, and then the recession of phalanges 4 and 2, and they kind of serve almost, they're like little dew claws, and those will allow for extension and contraction of different areas of the wing, so you can manipulate the feathers between the flight feathers and the primaries. So it's unlikely that these structures, they are so similar, it's very unlikely that they would have evolved independently in each species. Often when you're looking at structures that share a similar or the same complex bone structure or a unique body plan, it's often a case where you have a shared common ancestor. Kind of making things a little more interesting and complicated are analogous structures. So these are structures that have similar functions, right? They even look pretty similar, or they may look somewhat alike, but they are comprised of different structures and do not have a common descent. Thus, they have different evolutionary origins. And the great example are wings. So looking at the actual wing structure, or wing component itself, not the forelimb. So the forelimb is homologous. The wing itself is analogous. You look at the wings for insects, birds, and bats, they all evolved independently of one another. And you can actually see there's obvious differences in their structure. So with insects, the wing is comprised of this thin membranous layer that kind of stretches outward from the body plan. With bats, the wing is comprised of flaps of skin that are kind of stretched across and between the phalanges and extends outward from the forelimb. With birds, they have the extension of feathers from the forelimb, and those feathers are what kind of constitute as the wing. So with these structures, they evolved independently, or they were able to evolve independently in different organisms because the organisms lived in similar environments or they experienced similar selective pressures. We say they lived in similar environments, referring to they either occupied similar habitats, they utilized similar resources, or there were similar climates. Whatever the case, again, they lived in a similar environment and or experienced similar selective pressures. And this process is called convergent evolution. To converge means to come together like two lines meeting at a point. Convergent evolution is when unrelated species from different regions around the world converge or begin to look alike when they are found in similar environments. And a great case of this is these little guys here. So your pangolins, aardvarks, armadillos, and anteaters. A lot of people make the mistake of thinking that they are closely related because they look similar to one another. There's a lot of similar looking morphological structures, right? They all have these long snouts. They all eat ants and bugs. They have big claws. They have long tongues. So a lot of people hear about that or see that and they're like, oh, they must be closely related. None of them are related to each other. I think it's the pangolin is actually more closely related to the sloth or something. But they all look alike and have very similar structures because they evolved in similar environments and under similar selective pressures. So they all utilize similar resources, right? They all eat ants or termites. So they kind of allocated development of their sensory organs so that they could better find termite mounds or ant colonies. Their long tongues are used to access those colonies. The claws come into play with defense. Some use them in courting behavior. A lot of time it's used to actually dig up the earth to access the ants or to kind of tear apart large termite colonies. So because they're accessing the same resources and they live in similar environments, they have these similar structures, but they are the structures are at the structural level not the same, and they are not from a common ancestor. So sometimes organisms have structures that seem to serve no apparent function whatsoever, but they are homologous to useful structures in other organisms. These reduced or non-functional structures that some people like to call evolutionary leftovers, they're known as vestigial structures. And I like to think of vestigial structures as more as evolutionary footprints because they're remnants of a distant ancestor. And they provide evidence for evolution because they show structural change over time as well as common descent. Humans have a good number of vestigial structures. So the kind of famous one being your veriform appendix, or just the human appendix. In human populations, it serves no function except for becoming inflamed, infected, and then causing you to need surgery. But In other mammals, like horses, it actually serves a crucial purpose. It's necessary to aid in digestion. And we, at one time in our evolutionary history, we did utilize the appendix. So early hominids utilized the appendix was functional in early hominids to digest raw meat. But because we don't eat raw meat anymore, it's become non-functional. Another example are your auricular muscles or the external ear muscles. These are comprised of 3 different muscles. You have the superior auricular muscle, anterior auricular muscle, and posterior auricular muscle. Now this group of muscles, again, in humans, they're largely non-functional. But in many other mammals, they are crucial in producing ear movements. And by moving the ear, that plays a role in sound localization and can even serve function in the expression of emotion. Again, they're largely considered non-functional in humans. And Darwin actually proposed that humans kind of compensated for this, or the reason why these muscles are now non-functional is because they allowed for sound localization by just repositioning their head to better receive certain sounds. So that kind of compensated for the loss or gave reason for kind of eliminating the need for those muscles. There are cases, though, through repeated effort where people have regained the ability to wiggle their ears. I am not one of those people, but I also have not dedicated days of attempt for such feat. You are more than welcome to try if you want. Another example is the human tailbone or your coccyx. So between stages 14 and 22 of human embryogenesis, you can actually observe a tail-like structure that is then later absorbed in the embryo, except in cases when it's not, which sometimes happens. And that's rare, but it does occur. In other mammal species, though, or even non-mammal species, we see the preservation of tails. And in some species, it provides a function for stability or balance. Sometimes it's support, or it's used in locomotion or movement. So it becomes non-functional for humans, but it's functional in other species. One that a lot of people don't consider or forget to consider as a vestigial structure are wisdom teeth. So they were important in our ancestors. So early humans, the wisdom teeth actually allowed them to chew nuts and tree roots without hurting or causing damage to different mouth structures or the actual muscles of the mouth. But now they don't really serve quite the same role. So I'll talk about a few in other organisms. One that is often talked about are pelvic bones. So in most animals, the pelvic bones are needed in order to move the lower or rear set of limbs for the purpose of locomotion. Now, in some species, such as your whales or snakes and legless lizards, these limbs don't exist for the most part, but they do have vestiges of them. So despite a lack of need for actual limbs, you see pelvic bones in whales, your legless lizards, and snakes. They're very small and reduced in size compared to their other animal counterparts. What, Figaro? Stop! It's not your food time. No. They're quite reduced compared to their other animal counterparts, but they do exist. And for a while, there was this question of, okay, so why do these species that lack lower limbs or don't need pelvic bones, why do they have pelvic bones? And furthermore, these are pelvic bones that are actually functional in other organisms. So what's the purpose? And there was this thought that went around for a while that, well, maybe the pelvic bones served some sort of function. Maybe they help to support the whale's reproductive anatomy or the reproductive anatomy of the snake, but there are other structures and many other structures that are better suited for that task and actually kind of serve suite to that. So it doesn't make sense that would be that they're serving some secondary function. The only explanation that makes sense is that these creatures evolved from a common ancestor, along with all other tetrapods. And those early tetrapods had pelvic bones, and that's because it was present in the common ancestor, it was then present in these species. Over time becoming reduced in those species that don't necessarily utilize the pelvic bone or have need of it. Sorry for the squeaking in the background. Figaro found his squeaky toy. He's getting very annoyed because it's closing in on his food time. He gets fed at 5 or after 5 o'clock. So usually the hour before that, it's chaotic kitty nuisance festivity time. It's lovely. Anyway, another vestigial structure is the third eyelid. So if you look in the mirror, you look at the corner of your eye, you'll see the pink tissue right in the corner of your eye, and then the white of the sclera, right? Right in between the two, you'll see this very thin membrane or tissue. It's translucent, and again, it's very thin. That's called the semilunar fold, or the plica semilunaris. That is a vestigial remnant of the nictitating membrane. The nictitating membrane is the third eyelid that is still functional and common in many other species of mammals and non-mammals. This kind of nictitating membrane, it's used to move across the eye to protect it or to moisten it as needed. So you'll see it with your desert reptiles to protect the eye from desiccation. It's to moisturize in dry environments. Other species will use it to protect from dust or other particulate matters that may spray up into the eye. Sometimes it's used so that the actual eyelid itself doesn't have to close. There was that one notion that was kind of thrown around. We have no need for it. We have, again, the vestigial remain of it, and also the muscles that were associated with the semilunar fold, that pre-existing nictitating membrane. Those are also non-functional. We have no purpose for that. We have eyelashes and overproductive lacrimal glands. Another great example are your flightless birds. So most of your flightless birds, there's some great examples in New Zealand, but also penguins are examples, and ostriches. So with penguins, they don't fly, but they still have these wing-like structures. They use those for swimming. ostriches, they again also don't fly, but they have kind of these large vestigial wings that are not used for flight, but rather for courting behavior and for thermal regulation. And they actually will use it to help them cool down. But then you see these little guys. So these are kiwi. They're New Zealand native, and they have very, very tiny vestigial wings, very small remnants of actual wings. And a lot of people considered, for a while, didn't even consider them wings, but when you look at them, you see the same complex bone structure that's found in the forelimbs of functional wings. Again, everything is just greatly reduced. Now, the reason why a lot of these New Zealand birds, or you could say one reason why New Zealand has so many flightless birds, is that before humans came into play, like 1,000 years ago, there were no land mammal predators for birds. Rather, their predators were birds of prey. So, because most of the predators existed in the sky rather than on land, a lot of these kind of birds adapted to living on the ground, foraging on the ground, nesting on the ground, and all collectively living there. That was kind of provided more safety for them. The forest floor became more of a safe haven than taken to the sky. That's changed since people have arrived. Since that time, the floor has become more of a place of threat and peril for these birds, unfortunately. Another reason why they've kind of remained flightless, or you had this kind of a large population of different flightless species, is that flying takes a lot of energy. So because the ground was so safe for these species, birds could actually save energy by walking. There's a debate. I haven't read up on it recently, which is a shame, but I know at one time there was this debate as to whether these flightless birds, like the kiwi, evolved from a flightless ancestor, or whether it was rather it lost its ability to fly. I think it was more that it kind of over time it lost its ability to fly. There was no need for it to fly, so you had the reduction in that structure. Because you look at the structure, the complex structure of the bones. That structure is the same as the forelimb structure in functional wings. It even has this little tiny claw at the end of it. It also has the same attachment sites, the same muscles originate, and the same insertion points. So the whole, looking at the structural morphology, it makes sense that this was something that existed. So they started off with functional wings, and then over time lost that ability because it was no longer necessary for them. All right, so I'm going to stop this part of the lecture at this point. So this will be the end of part 1. You can follow the rest of the lecture in part 2. We'll start up with comparative embryology and then finish up the rest of the different avenues of evidence for evolution.